Welcome back, biology students. Today we started our combination of blue, gold, and Viking, and it was a little bit hectic, to say the least. I think we did okay, but I definitely need to work with my managing different pieces along the way. So I thought I would make a recording of our agenda rather than the video. So today we spent the day modeling meiosis, so getting our hands on some materials and moving them around. At home you can use any material you want. Pipe cleaners work great. You can use beads as centromeres, but you need to be able to show sister chromatids, homologous chromosomes, tetrads, and you need to move a cell all the way through each of the phases of meiosis. Okay? So in Google Classroom you have an agenda slide, or I'm sorry, you have the requirement sheet. Notice you need a diploid number of 6, right? So this is 6. 1, 2, 3, 4, 5, 6. Notice each has a single that was copied and it's connected to it and it appears like an X. Okay? So these are 3 tetrads. Purple comes from one parent, white comes from the other parent. So in your model, you need to show homologous chromosomes, those that are of the same length, are similar. You need to show sister chromatids, the white pipe cleaners are identical in length and content. You need to show the tetrad, the coming together of the 4, and the crossover, where they might swap some genetic information. You need to be able to follow those chromosomes as they move through meiosis. So, on to the next slide. Most of us modeled using markers on our tables. Now this model doesn't have necessarily 3 different lengths. I need to see that these 3 chromosomes are different, right? So you need a long, a medium, and a short. I have 3 from mom and 3 from dad. Notice in metaphase, they're still in that X form. And that X form is 2 pinks together. 2 blues are together. It's not a pink paired with a blue. They would not be sisters. I still see crossing over had occurred because these blues have a little bit of pink to them. Okay? You're going to take those homologs, pull them apart during anaphase, and then recreate 2 new cells during telophase. Prophase, nuclear membrane is going to disappear. Sister chromatids are now lined up at the equator during metaphase 2. Anaphase 2, sisters pull apart. So this should be 2 pinks pulling apart, 2 blues pulling apart. It should not be a pink and a blue being pulled apart. Okay? And this will be divided once again, and in the end you'll have 4 chromosomes, or 4 cells, with 3 chromosomes each. Okay? None of them should be alike. If I were doing this, say, on my whiteboard, I might grab some shape tool, circles. Okay. And so this would be 1 cell. And I can make 2 cells. So prophase 1, metaphase 1, anaphase 1, and my telophase is probably going to be a little bit smaller because it's 1 cell that, all right? So you're going to do this yourself. You need to label everything, so check the requirement sheet. In here, to start, you have 3 chromosomes. Let's see what I have to play with. So you're going to have 3 chromosomes of 1 color. You're going to have, and then we need to grab another color to be the other parent. So if I'm using blue, I have a blue long, a blue medium, and a blue short. And I want these 4 to represent a tetrad, right? So I should show some tangling up where they get switched apart. Okay? Metaphase, you're going to follow these same chromosomes all the way through. Okay? When you're done, you should have, with meiosis 1, you should still have a set of 3 X's to represent 3 chromosomes. They're not necessarily 3 blues or 3 reds. It could be combinations of. I'm just drawing this for simplicity's sake. Okay? So at the end of meiosis 1, you should have 2 cells with 3 sets of sister chromatids. Okay? At the end of meiosis 2, you should have 4 cells and they will only have one of each sister. Okay, so they're going to have either the blue or the red, long, medium, and short. And then they'll either have, if they got the long blue, red, they maybe have the short and the medium. But now you're only going to have 3. Okay. Okay. So 2 short. So these two need 2 medium blues. Hard to tell my random again. So 2 medium blues. Okay. So each at the end of meiosis 2 will have 3 individuals. At the end of meiosis 1, they have 3 sisters paired together. Okay? So that's the activity that we are doing today. We are modeling meiosis. So go ahead and get started.